share some of the exciting work that Fozzie is supporting in my lab, and why we're so passionate about pursuing this particular line of work. So start off with the graph here on the left. And the reason it's particularly relevant to me is because my mom happens to be a statistic on this graph. So she has lupus, which is kind of your classic autoimmune disease. So these are diseases where the immune system, which normally attacks things like bacteria, viruses, things that aren't you, for reasons that we don't totally understand, start attacking self. So in her case, skin, kidney, etc. And as you can see, the prevalence of diseases where the immune system is mistakenly attacking what should be friendly is increasing at an exponential rate. And this is really since around the 1950s. And it really can't be explained by a lot of confounding type of things that sometimes can contribute to graphs that look like this. For example, the fact that doctors didn't recognize those diseases or they were being underdiagnosed. I think it would be factual to say that diseases of the immune system, like lupus and many of the other ones listed here, are increasing at an exponential rate since the 1950s. Now, if you direct your attention to the right, the next logical question will be, well, is this happening everywhere? And the answer is no. So if you look at the world map here, the places that are green, the greener they are, they have a higher prevalence of another very common autoimmune disease, rheumatoid arthritis. This is where the immune system errantly attacks connective tissue like the joints. And you can see it's not a random distribution, right? Not the whole map is getting green. And it really is only in industrialized countries. And the best examples are the two teal ones here, Nigeria and South Africa. And here, what the authors have done is they've split the prevalence of rheumatoid arthritis by whether or not the residents were in urban industrialized areas or in rural areas. And so you can see it's really not the country per se, but rather we think is an effect of industrialization. So fast forward about 20 years, and I remember my wife calling me when I was in the hospital, and she had taken my firstborn son to do peach picking at Bishop's Orchard. It's a local kind of pick your own kind of farmer's grocery store with angioedema. And so my son now is a statistic of the graph on the left. And you'll see it's very reminiscent of the graph where my mom is a statistic in the previous slide. And this graph from Tom Platzmills very nicely shows that allergic diseases, so all allergic diseases, are also increasing at exponential rates, and they also seem to be occurring kind of post-industrialization. And if you think about it, I mean, there are really a lot of similarities between the immune system errantly seeing yourself, which it shouldn't see as something dangerous, and seeing food, which it shouldn't see as dangerous either, as perhaps having a common set of problems. Likewise, if you look on the graph on the right of the world, the same places that allergies are occurring in at the highest rates are the same places that autoimmune diseases are happening at the highest rates. And they tend to be places that are industrial. So if you look in South Africa, for example, the same trend will hold, where it's not that everyone in South Africa has more allergies, it's those who live in industrial areas. And so a lot of, this is kind of epidemiological data that people have been looking at for a long time. And the natural question is, well, why is this happening? And for a long time, the paradigm has been what's been referred to as the hygiene hypothesis. And so maybe the simplest way of thinking about this is just that now we're exposing diseases in the absence of essentially being dirty all the time, right? Being too clean, a disease of being too clean. And this piece came out of the PNAS journal a few years ago. And what it asks the scientific community to do is to think about other kind of paradigms that may be not mutually exclusively, but may also be helpful in trying to explain why these diseases are increasing so rapidly. And it really asked the field to think about alternative explanations, because in pursuing experimental lines coming out of this paradigm, the fruit of that has been probably maximized. So a real quick primer here on kind of the central issue at hand and our approach to dealing with it is to understand how the immune system decides to engage and remember something that it sees. And we think that understanding this kind of simple biology may unlock powerful new paradigms for understanding what might be going on. So on the left is the examples I gave you with my mom and my son. So with my mom, self-protein, the immune system sees that as not an enemy. And with my son, in general, we see food proteins as not an enemy. So in those cases, the immune response, when it sees something like this, leads to a tolerant state. So it's not a state where, the immune system comes out guns blazing. It's kind of a mutual appreciation that this is not dangerous. On the right-hand side, and I used here just as an example that we can all relate to, would be an example of something that the immune system would want to remember, because its job is to remember these things so it can defend you against disease. So a good example here would be spike protein in the COVID virus and in the COVID vaccine. So if your body were to see spike protein out of context, just the spike protein, it would treat it like if it saw a self protein or a food protein. What makes the spike protein immunogenic, what makes it stimulate an immune response that comes out guns blazing, is the addition of another piece, which in immunology is called an adjuvant. And if it's the virus itself, it's kind of the other viral parts. And here this work of Ruslan Metsutov has been seminal in transforming the field in understanding the rules of adjuvants and in terms of stimulating immune responses to pathogens. So the virus contains parts that stimulate the immune system, and these receptors are all worked out, that then lead to an immune response where the immune system remembers this as being bad and will engage it. In a vaccine, we simulate this by adding other compounds that have adjuvant properties. So this is a really important concept to take away from today as kind of a short biology lesson. But the way we were thinking about this very simple principle was instead of allergies being a disease of absence, would it be possible that it's now a disease of gain, so that something happened as a result of industrialization that's shaping immune responses inappropriately? And so what we've overlaid here on Tom's very nice graph here are the coincident introduction of other substances into the modern industrial environment. So for example, aspirin was introduced around the turn of the century. And then with the industrialization of food processing and food preservation, food preservatives, especially oil stabilizing chemicals, were added into foods around the 1950s. And then ibuprofen was commercialized in the 1960s, in addition with many other drugs that now would be on shelves in places like pharmacies that would be obviously of much higher density in industrialized versus non-industrialized parts of the world. And so we made a very simple hypothesis, which is that, is it possible that what we've done is add something into the environment because of industrialization that has this adjuvant property that's now transforming something like egg, peanut, that was we used to tolerate, into something that now we come out guns blazing and we cause food allergy, potentially autoimmunity, and other diseases of loss of tolerance. So the way we tested this, and this is to a testament to kind of the vision of Fozzie, because this is kind of very high risk, high reward kind of work. We actually went to the FDA website and pulled their food additive status list, which anyone can do, and went through it and picked out ones that would be commonly found in foods that have a high rate of food allergy in the United States. So things that you would add to commercially processed peanuts, eggs, etc. In addition to this list, we also decided to look at things that we can that we give our children. We know, for example, now that there's a kind of a vulnerable period where food allergies are likely acquired, and they tend to be in kind of the early parts of development after birth. And so these Tylenol and ibuprofen, for example, are ones that are commonly used in kids, and that the American Pediatric Association use in kids for one reason or another. And so we asked the very simple question, and I'll walk you through these graphs. This is probably the only piece of data that we'll show today, but we'll walk through it together. We asked whether or not, if we gave a mouse egg protein and then simultaneously gave it ibuprofen, indomethacin, Tylenol, ketoprofen. There are many, many other ones that I'm not showing here, but these are the example that I gave you on the right. So these are examples of things you can get as over-the-counter meds. Would we be able to make a mouse allergic to egg protein? And so in the open circle is where we just gave the mouse egg protein. And what we're measuring here is the amount of IgE, which this audience knows is what develops. This is the immune memory that develops to food proteins that then lead to anaphylaxis. We're measuring the amount of IgE in the blood of the mouse. And you can see if you give just the egg protein, nothing happens. And this is exactly what you would expect, right? In the absence of an adjuvant, there's not an immune response. If we give a positive control, and so this is just to make sure the experiment worked, and here we used aluminum salts, which is commonly used in this line of research, you can see that the mouse nicely makes IgEs specific to egg protein. What was astonishing was when we gave it doses of ibuprofen that the FDA allows to mice, we get a equally robust formation of memory to egg protein of the kind of anaphylactic flavor IgE. But this wasn't true for all NSAIDs, right? So a cousin of ibuprofen, a very similar cousin in terms of its structure, indication, and use, it would be ketoprofen, for example. This ketoprofen doesn't lead to that response, and neither does Tylenol, but indomethacin does. And as a sanity check here, if you were then to challenge the animal that has these antibodies with egg protein, like they might encounter accidentally in foods, they develop an anaphylactic response. And in mice, this is measured as a very, very rapid drop in body temperature because of the angioedema and vasodilation that happens as a result of shock physiology. And so you can see the mice that develop the IgEs to egg protein are the same mice that then have this anaphylactic response. So on the left, just to summarize these two graphs, we essentially found that some NSAIDs, like ibuprofen, but not others, were sufficient to be adjuvants that could shape the immune response in a way that led to food allergy. On the right-hand side is that food preservative that I had put up on Tom's slide earlier. And this is tert-butyl hydroquinone. It's one of the most commonly used preservatives to enhance shelf life of fat-containing substances. So you can find it in most things. And we did the same experiment here, where we asked, could we use this compound with egg protein alone, and would that compound be an adjuvant and lead to anaphylactic response? And it did. And consistent with an anaphylactic response, the mice that were then challenged with egg protein had mast cells that degranulated their contents, as would happen in an anaphylactic response in humans. So the kind of the summary of these data slides is that we identified industrial xenobiotics, so food additives and over-the-counter drugs that could have adjuvant properties that could lead to loss of tolerance to food proteins in a way that led to food allergy disease in mice. One of the kind of great benefits of working in FOSSI is that a lot of the questions being asked here have kind of deep biological implications. And so oftentimes, while the projects are wholly devoted to solving food allergy, unexpected things can be discovered that are quite meaningful. And this is an example of one of them. And I wanted to highlight folks that did the work of the graphs before. This recent paper out of our lab as a result of that work. This is Anna Eisenstein. She's now a new professor in dermatology at Yale, who was a postdoc in the lab at the time, and Brandon Hilliard, who's an amazing postbac, who's now an MD PhD student at University of Colorado. But you remember I told you that it wasn't just all NSAIDs could lead to food allergy in mice, some but not others did. So we ended up working that out. And in working that out, we discovered a new property, a new kind of mode of action of some NSAIDs but not others. And it might explain why we've observed for a long time as clinicians that, the same NSAID properties amongst people lead to some very difficult to explain differences in outcomes. And so just as an example of what happens when you start probing at these deep kind of biological questions. So I think where we're headed, and this will be the last slide, so happy to chat about this work, is to know, we really think we're at kind of the tip of the iceberg here. So we've screened some dozens of FDA food additives in the way that I kind of described. I think the first question is, is it generally true that industrial chemicals are causing more food allergy because of their ability to act as adjuvants? And obviously these studies are done in mice under kind of highly contrived experimental settings. And so it will be important to do clinical studies to estimate the effect size if this biology in fact can be operating in humans. And that's also work that FOSSI is supporting with Dr. Gary Sofer, who's one of our allergists here at Yale, and Anna are doing a clinical study in allergic children to that end. From a biological perspective, while many of the properties of the adjuvants that lead to responses that are protective for viruses like COVID, for example, are extremely well understood, their molecular features, how they work. The same is not true for adjuvants that lead to responses for defense, not against viruses and bacteria, but the type of defenses that can lead to maladaptive diseases like food allergy. So defense against, for example, parasites and fungus would be the physiologic example. And we think that by mucking around in this space, we may be able to identify whether or not there would be the same kind of, in the end, fairly simple rules for defining what makes an adjuvant of this particular flavor to lead to that, this allergic type of inflammatory response. And then obviously, because as I kind of insinuated before, I'm a human doctor as well as a mouse doctor, and living with a food allergic kid. How can we use this information then to affect positive change towards getting rid of this disturbing trend in the increase in food allergies? And so with that, I'm very happy to take questions and hear thoughts about this work. And I'll stop sharing my screen.
Wow, thank you. This was exciting. And your mother must be proud of where you're taking it now from her disease, and now you're helping cure your child as well as her. So that's impressive. And I think that type of drive really makes a lot of change. And this topic is so relevant because when I talk to some people with food allergies, what I hear the most is, why is food allergies increasing over time? And this is kind of a look into it. And there was, everyone always talks about the hygiene hypothesis, but I love that quote saying time to get, stop blaming just hygiene hypothesis and look at other things. And so thank you for looking at other things and thinking about it. And there's lots of questions coming. And I know sometimes hearing this is slightly overwhelming because you start thinking, what can we do? Right? We have this home, you can walk to the pharmacy and you can get all these medications, you're trying to do the best for yourself, for your children when they have fever, bringing things into your home, and what you can control and what you can't control. So one of the questions is, what would you kind of recommend from a standpoint of, we don't have all the answers right now, but what can we do now to kind of make a difference in our health?
Well, I think I guess I'll say a couple of things. I think the first part, as I mentioned, these are highly contrived systems in mice. And so a couple of those features in the contrived system, I think, probably matter a lot. One is the fact that they're genetically identical and of one kind of genetic makeup. And so obviously allergy has a strong genetic component. And so the susceptibility to allergy is not the same for everybody, for example, just based on genetic makeup alone. The other aspect is that mice don't generally eat eggs. And so, for example, seminal studies like the LEAP trial very nicely illustrate to us that introduction of food proteins in certain developmental windows can shape the immune response to this tolerant kind of response. And the same thing would happen in our mouse model, which I didn't show. So for example, if they were pre-fed egg before, and then you tried to sensitize them to egg later with these compounds, and then it doesn't work. So we're not breaking the rules of oral tolerance as we understand them and have been clinically borne out. So they're just to kind of frame the discussion. I think in terms of what to do, I think, the first part is to understand the biology, which is, as you had mentioned earlier, Chris, a major focus of FOSI. And then once we really, really understand the biology, obviously to look to see what extent this is operating in humans. So doing clinical studies to evaluate effect size. And in the absence of those things, especially from a clinical perspective, it's hard to make hard clinical suggestions. That said, it should be intuitive, I think, to maximize to the extent possible. For example, buying locally sourced food, trying to avoid, if possible, and within the constraints that one has with their means, to only put things in your body that you think would have the kind of highest health content. And that's not always easy to do, but I think it's nonetheless worth striving towards. And then, at the same time, trying to avoid things that in various other settings we know to be correlative with other kind of bad health outcomes, right? So we know, for example, that highly processed foods can lead to other bad health outcomes. And so it's not really that big of a stretch to think that consuming highly processed foods might lead to this bad health outcome. So I'm not trying to be dodgy here, but just to speak in generalities.
Yeah, it's complicated, and there's a whole environmental impact to take in account that's hard to take in, right? It's nice and controlled in the lab and we can see it, and how does that go when you're, a little old, a little mature, maybe we're exposed, maybe weren't, and get these medications. And not everyone is going to react the same,
right,
because of the genetic profile and the environmental profile. So it is challenging, but it's important to think about, because you think about this is for food allergies, but it probably impacts many other areas as well. And I'll just go back to Michael Pollan's books, which I read a very long time ago, about like eat real food, nothing with more than 5 ingredients is key. But it's hard, I brought my kids skiing this weekend. You go to the lodge and, it's chicken fingers, French fries, and candy is out there. And that's about all they have to offer you. So it's not an easy thing to do, but me personally, I've tried to avoid lots of plastics in my household, try to, as much as possible, preservatives, and do little changes like that. But it is it's challenging in our world, but something to consider. So let me just, someone asked about you, and this is a hard question to answer. Would you consider mothers in the hospital giving birth dosed with painkiller meds as potentially contributing to the allergies of their babies and breastfeeding?
That is a great question. We thought about the same thing. You can imagine we also thought about, during our childhood vaccination schemes for our babies, they can get quite fussy, and ibuprofen and Tylenol are very soothing for the children when they have to get this. I think, Chris, you put it best. I think there's lots of moving parts. It has to be kind of the right combination of genetics plus the right dose probably of the thing, given in some kind of proximity to the food that maybe you were or were not exposed to one way or another. And while it sounds a little bit fatalistic that then all the stars would align in a way that produce a disease state, many diseases have this property where many of the quote stars have to align, right? You have to have a certain kind of genetic disposition, and things have to happen to you in a certain way. I think the important part is to understand what those stars would be that you have to align, because then you can start having productive conversations about what one should or shouldn't do. I think it's too premature to say, let's not give a woman who's had a C-section some painkillers and send them home with NSAIDs afterwards without knowing truly what the clinical effect size of this would be. Obviously, NSAIDs are things we all take over the counter all the time, and it's not like everybody has food allergies. So that from an intuitive perspective should be obvious that there clearly need to be other ingredients here that need to be in place.
Yeah, no, it's a nice window, but there is more science that needs to be done before you do that. I mean, we're both physicians, I don't give out medications or to whether my patients or my kids unless they really need it. And I
Likewise.
talk to them about these risks. And I'm one who doesn't take Tylenol, ibuprofen unless I need it. You only, so just minimalizing and using it as you need it. And something like that, you'd have to talk to your doctor, but typically they're needed for women at that stage. Great talk and fantastic findings. Have you checked whether the same xenobiotics, and when we talk about xenobiotics, we mean unnatural chemicals, right? Can act as adjuvants in autoimmune context?
We are looking, let's put it that way. The expectation would be it's possible. So just as an example, many of the mouse models for autoimmune diseases like multiple sclerosis, rheumatoid arthritis, and for example, lupus even, are induced in mouse models with antigen adjuvant combinations. So they can be done in mouse models. And the question is, what else can do them other than pristane and Freud's adjuvant and these other adjuvants that we use? But the screen in mice would be the same, right? Can you cause antibody responses of the kind that would be directed towards viruses and bacteria, the same kinds that are a problem in lupus and rheumatoid arthritis in mice models? That it's a great thought, and yes, absolutely.
And so that just brings me to think that this is, one thing that we're not talking about that we're also looking at is the microbiome. And the microbiome plays a big role in this as well. So when you talk about giving birth and all this, there's lots of areas of interest and research to do with the microbiome and what's passed on to the baby. But as well as, and this is something we look at in many fields, including oncology, is how are these drugs metabolized by the bugs in your guts, and what influence does that have? So there's other things besides just straight this that play a role. And one of the studies that we are looking at now is seeing how those bugs break down the drugs and cause an influence in the immune system as well. So it's complex body system that we're solving, but we're going to move ahead. So one more question, the TBHQ, how do you think that drives sensitization to OVA? Lots more questions.
So I guess we'll have to get a little bit technical here, but basically TBHQ activates the xenobiotic sensor NRF2. So in the last schematic, we didn't go over a lot of the details. it turns out that some of the NSAIDs also, the NSAIDs that led to sensitization to egg allergy also activate NRF2. And NRF2 is this, it sits in the cytosol with chaperone protein, basically waiting to see excess amounts of electrophiles. And many xenobiotics are electrophiles. And so it's a built-in kind of sensor to make sure that there's not a crud inside of the cell. And when there is, it activates a program to detoxify that crud, to detoxify the xenobiotics. And so TBHQ is a known NRF2 activator. And to make a kind of a very long story short, what happens when the cell is engaged by TBHQ is this is also activated. It releases one of these damage alarmin signals, and it's this alarmin signal that then engages the rest of the immune system. So we think we have a pretty good handle on the molecular events going from the mouth to then the immune response.
Yeah, and we do have some scientists on the call, and you can always email me directly as well, and we can talk more about it. Do you have any preliminary thoughts on adjuvants may or may not influence the preborn infant through breastfeeding after birth, which we covered a little bit there with the painkillers?
Yeah, and I think I think maybe the question is also directed at kind of intrauterine events. I think Chris is, your point is a really, really good one in terms of drug metabolism, because these are, xenobiotics are basically drugs, and a lot of things affect how they're metabolized, and then a lot of things then impact whether or not they go across the placental barrier. So a lot of moving parts.
Could you comment on the critical immunological changes that contribute to the development of food allergies in infants, especially in the first 4 to 12 months of life, and how early introduction of some common foods can prevent the development of food allergies? Which goes along with our second question as well, talking about maintaining a healthy skin barrier as well, and how that can develop, which goes along with the early introduction.
Sure. So in the first question, I think maybe the simplest way to think about it is that the GI tract, the gastrointestinal tract from the mouth to the anus is, it's really, really important, because it's where we harvest energy from the environment, right? So if you don't eat, it's a problem. So the system has to balance and preserve at all costs the ability to extract nutrients, micronutrients, essential nutrients, and energy from food. At the same time, Chris, as you nicely pointed out, there are more bacteria living in there than there are cells of you. And so we have to also make sure that environment stays controlled from, for example, if Listeria hitchhiked on your spinach, or any number of pathogens get in there, that the system is also aware enough that there's now a problem inside of the place where it needs to extract food to keep you alive. And so the GI immune system is special in that regard. So we talked about the concept of tolerance. Oral tolerance is probably at the heart of the first question, which is that if you teach the immune system at an early age that this food protein is not dangerous, then it shapes the memory of that child's immune system to know that, well, when they see this is not a problem. I hope that addresses the first question. The second question about this idea of epicutaneous sensitization or sensitization through the skin, Chris, as you know, is something that Fozzie is also funding from my laboratory. I think that one way of thinking about it could be that, if one is always getting exposed to an otherwise inert food protein like egg, in the context of something really bad, like your skin's messed up, or your gut's messed up, or any number of things that other parts of the sensory system would perceive as aversive or dangerous, and that you want to remember it. The immune system has this funny feature of remembering proteins. It has a hard time remembering other things. And so the protein then becomes kind of a proxy for whatever the else bad is, and it could be skin injury. And the way that could connect and its implications are also kind of deeply interesting as a kind of a conditioning paradigm. And Dr. Metzdoff and Dr. Florsheim, another Fozzie investigator, published a very, very nice review kind of exploring those new paradigms of thinking about how to shape immune memory in kind of ways that people have not talked about before, or if they have not kind of flushed out in this kind of detail. And so it's a very long answer to the short question, but I think it's complicated, right? Because a lot of babies will also have non-atopic skin problems, and those aren't babies getting food allergies. So what's the difference between one kind of skin injury to another kind of skin injury that makes one more likely to get food allergies? And those exactly are the type of questions that we're addressing.
Yes, and it could be there's a more genetic component for those developing eczema over others, which leads
Sure.
to the development of food allergies. But you know what, the way we think about food allergies and the development of them is what, you know, there's different modes of sensitization, meaning different ways to develop food allergies. There's not just one way, which makes this slightly challenging, because if it was just one way, it'd be much easier to solve. But some people might get it through the skin when they're young. Some people may get it through the gut, and there we are examining that crosstalk between the gut and the skin. We are looking at, someone else, we're looking at chemicals on the skin. We do look at that as well, and what chemicals are causing the skin reaction, potentially leading as well. We have some interesting data, and actually Dr. Sokol next month will actually be talking about that on March 27th of that data, looking at what triggers the skin to development of food allergies. But Dr. Mizitov also published this paper on food quality control, which is what it comes down to, is that the body is, you know, you have to think of the gut as something that's exposed to the outside world. And it's really the only thing exposed to the outside world, right? Your skin's a barrier, your lungs is gas exchange, if we take away all the bad stuff in our environment. And it had to develop a mechanism to detect what comes in and make sure it's safe or not safe. And if it was safe, to absorb it, and as nutrients, if it was unsafe, to get rid of it. And when you get rid of something, when your body gets rid of something because it thinks it's unsafe, it has to remember it. And it remembers it not only by the immune cell, but by the brain as well, you know, to make sure next time it sees it. And as Dr. Wang was saying, it's better at seeing protein than chemicals. So when you think about it with, you know, the preservatives, is they might see these preservatives or these organophosphates in your food every single time. And eventually, it usually doesn't happen the first time, it says, okay, we don't want this in our body. And it associates it with whatever food you're eating that has that protein, whether it be the peach that maybe, you know, was your son at the farm that happened, you know, because it has some chemical on it, or perhaps it was, you know, the preservative in the food that Dr. Wang talked about already. But we're getting to the end of the time. Thank you so much for joining us today, Dr. Wang, and really giving us a peek into what we think is some of the root cause of the development of all these allergies that are occurring as well. And I just want to thank everyone. You know, FOSSI is really transforming how we think about, treat, and diagnose food allergies, and all of you have really helped bring that by continuing to donate to FOSSI. So thank you all for that. We want to continue with these major breakthroughs as well, and we hope to see you on March 27th to meet with Dr. Sokol to hear more about her work on skin and development of food allergies as well. So thank you and have a wonderful day. Bye-bye.